Second. Okay, Charles Keller, welcome.
Yeah, thank you very much, Roger. So I'm Charles Keller, Scientific Director of cc-TDI, the Children's Cancer Therapy Development Institute, which is one of its kind. It is a 501c3 nonprofit biopharma spin out of Oregon Health and Science University. We do the R&D on the cc-TDI nonprofit side, but we wholly own Artisan Biopharma, which then does the drug commercialization, partnerships, and investment. So science has never been better, and we'd like to apply the state of the art of science to childhood cancer drug development. So since 1978, there have been 13 drugs intentionally developed that have reached FDA approval, so 13 in 47 years. For adults, there's 12 to 17 every single year. And so to balance that, we created this ecosystem of cc-TDI as the nonprofit R&D lab and Artisan Biopharma for commercialization. In the period of 10 years that we've existed, we've had a number of NIH and Cancer Research UK fundings, about 2 to 2.5 million a year. So we run lean. But we also managed to publish quite a bit, 66 papers in 10 years, and 3 of those papers in genes and development, science signaling, and nature medicine led to cooperative group trials. And so it's with this idea in mind that we're developing a treatment for a childhood brain tumor. The team to build this treatment include people both on the cc-TDI side and the artisan side. I'll just highlight on the artisan side, we have Dr. Tony Haight, who spent 30 years in pharma, most recently as head of the emerging therapeutics platform for AbbVie, which included development of antibody drug conjugates, and Cody Stiverson, who is an F-15 fighter pilot, but also did a lot of national security work that brings him as a perfect partner for operations. And he has all the heart in this because his daughter Addie was a childhood cancer patient. We have additional scientific advisors on the cc-TDI and artisan side, including Tim Brown, shown there on the right, a recently retired vice president of Genentech. So this is the essence of the problem. So this is DIPG. The white part is the midbrain. One's midbrain should be about the size of your thumb, but with DIPG, it becomes the size of a plum. And that's just too big for the skull. So this is usually young children. It's the only disease for which we say that it's 100% fatal. We don't give the parents any inkling of hope. The median survival is 7 months without radiation, 9 months with radiation, and there are no long-term survivors of this disease. So we're here to solve that with an antibody drug conjugate. And antibody drug conjugates really have had a very nice moment in the past few years. Daiichi and AstraZeneca introduced an antibody drug conjugate to the HER2 protein in breast cancer. And as you can see on the left, the orange curve was significantly better than any other antibody drug conjugate that had previously existed. Well, the reason for this is that the linker and the payload attached to this antibody are next in class. So to think a little bit about what was done by Daiichi for breast cancer to improve survival, they did so with an antibody drug conjugate for a protein that's found in very much across the body. As you can see on the left, the RNA expression and the protein expression are quite high. And this led to 16% of patients having to stop the medication because of toxicity. Well, if we find a target that's less common, so for DIPG, the IL-13 receptor, its expression is really quite low in most places. So it makes an even better target than HER2, which is already a breakthrough drug. To design the antibody drug conjugate, you need an antibody, you need a linker, and then you need your payload, here shown as the toxin. There are many considerations. The first consideration is that the tumor simply should have the protein that the antibody recognizes frequently in many patients' tumors, but also across the tumor uniformly for each patient. Ideally, an antibody drug conjugate acts a lot like an antibody and has a very long half-life, so it doesn't need to be dosed daily, but can be dosed weekly or even less frequently. And the payload, well, the payload has to be inherently active against the disease. So, in looking at IL-13 receptor alpha 2 as a potential target in this disease, diffuse intrinsic pontine glioma, a glioblastoma of the pons of the midbrain for children, it would, from our publications, appear that about 80 to 90% of cases are positive, and they're highly positive in at least 55 to 60% of cases. So that makes a good argument this is a good target. But the other important aspect to consider is that there's an adult market. And here I'm just making the point that melanoma has high levels of expression of IL-13 receptor alpha 2. So the pediatric indication, DIPG, the adult indication, relapsed melanoma. So just to sort of dial in a little bit deeper on the protein expression for DIPG, the tumors have a great deal of the IL-13 receptor alpha 2, but the normal brain does not. So we have some specificity. It's a fairly uniform expression. And I'll just mention as passing that this New England Journal Medicine article with a patient with a Lazarus type response to a CAR T against IL-13 uh, did indeed have the same target that we're building an antibody drug conjugate against. Well, an important aspect of any study is to ensure that if your antibody drug conjugate recognizes proteins on the outside of the cell, that in fact the protein is found on the outside of the cell. And so for DIPG across these three different cell lines, we validated that the expression is always on the membrane and not so much in the cytoplasm. So the drug can get to the cells that we want to target. Proof of concept. So the next thing we did was we built a proof of concept antibody drug conjugate for testing on the R&D side. And so that's composed of an antibody, in this case, an IgG1, a linker, which we're using one very similar to the Daiichi one, and finally a payload that's active in the disease. And so I won't dive into details, but I'll say that there are two commercially available antibodies that we could develop for this proof of concept on the R&D side. And the payloads that were available were tested against a number of different cell lines. And fortunately, the Daiichi-like topoisomerase inhibitors had activity in the single digits, tens of digits, or hundreds of nanomolar, but generally less than 1 micromolar. So we felt that was a good payload to have. We took some genetic model systems and ensured that the antibodies had specificity, that they only recognized the tumor cells that had the IL-13 protein expressed. And we constructed this antibody drug conjugate with these commercial antibodies, the linker from Daiichi, and the payload that we would otherwise want and have activity in this disease. And the very nice thing is that the antibody drug conjugate was high performance and even had better affinity for the IL-13 receptor than expected. So the testing of this in the Petri dish over a period of 6 days in vitro showed that the antibody itself had no toxicity, but the antibody drug conjugate had toxicity, but only to the cells expressing the IL-13 receptor. Now we have a very nice tool in the lab. We borrowed the quail egg shooter, commonly sought after in sushi restaurants, and we turned this into an animal testing platform that can be automated. So essentially, if you take the top off an egg, as shown with this quail egg shooter, drop it into a dish like this robotics ready 6-well dish. The yolk goes to the bottom, the embryo goes to the middle, then there's a membrane that sits on top that has arteries, veins, and lymphatics. It's very vascular, it's shown in the side cross section, and you can place tumor cells on it that express firefly luciferase and then test drugs and look at changes in luminescence as a readout of this animal xenograft testing, which instead of taking 10 weeks and $45,000 at a time, only takes 10 days and 35 cents per egg. And the testing, therefore, of these three different DIPG cell lines with either the antibody given in blue, which had no effect as expected, or the ADC shows efficacy of the antibody drug conjugate to this specific tumor. So we showed you the in vitro results, and these are the animal results. Of course, what we showed initially as proof of concept needs to be moved to translation. And through Artisan Biopharma, we are licensing from a major pharma an antibody with a very, very high affinity for the IL-13 receptor, uh, just 950 picomoles. It's already been validated by the pharma to have rapid internalization, so we can bring the payload into tumor cells, and it's patent protected. For the linker and payload, we're building those ourselves through the expertise of Tony Haidt, who's developed these in the past. So in terms of milestones for investment, there are three tranches. We're trying to get it to a total of 9.9 million to get to the IND stage. The first tranche, 174,000, second tranche, 319,000, and thereafter $6 and $3 million tranches. All right, so that brings us to the end of the presentation, and I hope I have convincingly shown that IL-13 receptor alpha 2 is a high-value target for a disease of great need, a uniformly fatal brain tumor of childhood. We developed the antibody drug conjugate that has strong preclinical activity, but we're also licensing from pharma the best available antibody on the market for this, and we're on a path to developing the medicine. And bringing this drug to clinical trials. Thank you.